Okay, we will invite Dr. Mahmoud
Professor
Musli.
Professor Mahiba, Professor Sulaiman, welcome for your session.
أيوة.
We invite Dr. Mahmoud Musli to speak about
Professor-
biologics between present and future. Dr. Musli.
ايوه اهلا وسهلا كيف الحال؟
الحمد لله.
طيب معلش I'm going to switch to my laptop, just give me 1 second. Okay, can you give me admin so I can share my screen? Can you see the screen now?
Yes, it's okay.
Hello?
It's okay. It's okay.
Okay, طيب.
Thank you. Thank you.
Thank you very much. Bismillah ar-Rahman ar-Rahim. I'd like to thank the scientific committee of this meeting for inviting me to speak. It's a real pleasure to be among friends and colleagues and to be involved in this very important meeting, which we are all very proud of as the first meeting to unify us as Arabic speaking IBD specialists. I've been tasked today with a topic that has to do with more of a historical journey through time, discussing how biologic therapy has evolved over the past 2 decades. These are my disclosures. We're going to talk about first the rationale for using biologics in IBD, and then the evolution of the timing of biologic therapy introduction in IBD, in addition to introduction of new forms of biologics into the IBD treatment paradigm. So as we have all heard over the past several years through multiple lectures and many scientific meetings, one of the most important observations is that Crohn's disease is a very time-sensitive disease. It's a disease that has many implications when the diagnosis is delayed. Time matters, as bowel can be damaged with time once the activity persists without any treatment. If you can see on the left side, these are the figures taken from the Lehmann index paper. When it was being developed, you can see how there's a window of opportunity where the disease can be ideally targeted and treated to prevent any damage to happen over time. So one of the important things to see on the right side is if that happens and we are able to prevent inflammation persistence during that time period, that curve of progressive bowel damage can be flattened and complications can be prevented with time. This is another figure taken from a from a French cohort study that basically followed a cohort of IBD patients over the span of several years. And you can see with time how disease that originates as pure inflammatory phenotype can progress into stricturing, then penetrating disease if inflammation and disease activity persists. And that, of course, ultimately leads to the need for surgery. Now, over time, historically, treatment of Crohn's disease has highly evolved and changed. Initially, in the 1960s and 70s, the primary mode of treatment was actually surgical intervention and resection, but with time, it was identified that that approach only is associated with high risk of morbidity and mortality and is not a cure. And of course, it's non-curative. Following that, antibiotics were introduced as treatments, was found to be non-effective. And then there was the approach of targeting the immune response through non-selective blockade of the inflammatory cascade that started with steroids and then evolved into immunosuppressants and finally biologic agents. This, of course, led to off-target side effects, which led to the drive to develop and manufacture more specific type of targeted therapies, i.e. get selective anti-inflammatory agents and anti-cytokines. Of course, we know historically that steroids is not effective beyond the period of induction and ultimately is associated with lots of complications, including adverse events to the drug itself, and does not really alter the overall long-term outcome of the disease in the future. One of the very popular drugs that was asked about in one of the earlier sessions, I believe in Dr. Majid's session, there was a question about how immunosuppressive, specifically azathioprine, is used. This is one of the drugs that initially was very popular in the 1990s, but more recent studies over the past 10 years have shown that despite an increase in the use of these type of drugs between 1978 and 2002, there was no decrease in the rate of surgical resection, i.e. that the drug does not really alter the natural course of the disease. It is now only used in very specific contexts, but otherwise, in my opinion, is not a primary mode of treatment. Of course, with time, not only drugs have changed, treatment goals have changed to mirror what has happened in the treatment paradigm. We are now focusing more on objective markers of inflammation and objective methods of evaluating disease activity, which leads us to the next slide, where we discuss and describe the different type of medications that are overall used. And I'll highlight here in the middle the definition of biopharmaceutical drugs, which is a medicinal product developed by means of one or more of the following biotechnology practices: rDNA, controlled gene expression, and antibody methods. Of course, there's a huge difference between chemically based drugs and therapeutic proteins, i.e. biologics, with regards to the way it's manufactured and the approval process for these medications as well. You can see here that these therapeutic proteins are made by harvesting the substances produced and secreted by constructed live cells, of course. It has high molecular weight, complex three-dimensional structure. It's produced by living organisms, therefore often heterogeneous. It's difficult to characterize completely by physical, chemical, analytical methods or bioassays, and it's prone to eliciting an immune response. So it's highly immunogenic. Of course, these drugs are very complex. They have a quaternary structure, which makes it very complex in its immunological characteristics and in its mode of action. Of course, these come from production cell lines, which are proprietary and not available to the producers. Of course, we know that with time, there has been the development of biosimilars, a topic that will be discussed later, so I will not be touching on this during this lecture. Of course, biopharmaceuticals is very different from low molecular weight drugs, which we have now highlighted. We know that biologics work really well in IBD. We have drugs like anti-TNF agents, infliximab, adalimumab, Pegol, and we have more recently developed biologics, natalizumab, which is rarely used these days because of certain side effects, but we have ustekinumab and vedolizumab drugs that have been already discussed in this lecture series. Of course, we heard about gaps in treatment, and we can all agree that there's significant gaps present in the treatment paradigm of patients with IBD. Of course, one of them is availability of the therapy, such as financial barriers and accessibility. And then there's what happens after the drug is given. So primary non-response, secondary loss of response, which is a pharmacokinetic driven phenomena, non-compliance, intolerance, and adverse events, with a focus on infection, metabolic derangements, and fear of neoplastic complications. Now, with time, the timing of biologic introduction has changed and evolved. Initially, it was used only after failure of conventional therapies for CD and UC, i.e. steroids and immunosuppressants. Of course, the role of these type of medications in this context is not really debatable. There's a growing body of evidence supporting this approach. Then came studies that supported the use of biologics in treatment-naive patients, i.e. immunosuppressant naive or steroid naive patients, and the results, of course, here are as well not debatable. Fistulizing Crohn's disease patients are also candidates for treatment with these agents, and following resection for Crohn's disease as well. It's also used as a rescue therapy for acute severe colitis following failure of steroids and for extraintestinal manifestations of IBD. So the first indication, failure of conventional therapies, of course, we have already discussed that this disease progresses with time, and time leads to complications. Now, if you would think the ideal point of introducing these agents is before any of this happens, even before the inflammatory burden peaks, then you can start with what happened historically is the introduction of this drug somewhat later in the course of the disease, such as the population studied in ACCENT. So in ACCENT 1, the patient who failed therapies to initial drugs were all randomized or introduced recruited into these studies to receive infliximab, which was the first anti-TNF to be studied in the late 1990s. So as you can see here, the majority of these patients are patients who have failed conventional therapy. They had a significant proportion had prior surgery, had long median disease duration, and were already exposed to 5-ASA steroids and partially to immunosuppressives. So results at the time were fairly impressive due to the lack of other therapeutic options. This was considered revolutionary at the time, with patients responding in a very short duration within weeks. You can see here that even the long term follow-up, a significant proportion of patients remained in clinical remission without the need for steroids. The same population, but in ulcerative colitis, underwent similar pattern of studying, and again you can see very similar disease demographics and duration of disease, and again these patients responded very well, and following this the drug was approved as an option of treatment initially for ulcerative colitis, then for Crohn's. Now, if you think about how time led to scientists considering introducing the drug early on in the course of the disease, you have to mention the landmark study SONIC. SONIC was a very known study in IBD where patients who initially were naive to immunosuppressives and anti-TNF were randomized into the study to receive either infliximab with placebo, infliximab with Imuran, or Imuran with placebo. The main aim was to study the benefit of using combination therapy to prevent immunogenicity and improve outcome of drug efficacy of infliximab. And as you can see here, a very different disease patient population was recruited, with only a smaller number of patients having received surgery in the past, a shorter duration of disease, and again, as mentioned, mainly immunosuppressive naive type of population. And then, of course, the results were overwhelming, with very statistically significant differences supporting the use of combination therapy for this type of patient population. A similar study was performed in ulcerative colitis, which is UC success, and of course the drug was deemed effective similar to Crohn's disease for ulcerative colitis, with some modest benefit of combination, not as pronounced as that for Crohn's disease. Now, it's very obvious that with these results, we'd like to push further and try to examine the benefit of the drug in a patient population that had just been diagnosed without exposure to any drugs. And that's where the study Step Up, Top Down by Professor Geert Hans was performed and conducted. Of course, these are patients who are newly diagnosed, no surgery, median time or disease duration is 2.5 weeks, none of them exposed to steroids, immunosuppressives, or anti-TNF. So it's a very, very newly diagnosed population of patients. Of course, here conventional step-up treatment was compared to early combined immunosuppression, what we have seen with the results of the study is that early aggressive patients who had received early aggressive treatment approach did fairly well compared to those who followed a conventional therapy approach, highlighting how early introduction of biologics in the course of the disease can be very beneficial. This was replicated by other studies such as CHARM and EXTEND and PRECISE, where patients with shorter duration of disease seem to do much better than those who have longer disease duration. This is a summary of all of these studies in both diseases using different approaches. Now that was the historical evolution of anti-TNF, but then 10 years ago, more just like Prof. Izzat mentioned, the pipeline was active with the production of many different mechanisms of action, drugs that acted in different ways to treat Crohn's disease and ulcerative colitis were studied, and eventually some of them were approved as viable treatment options. Now, we'll talk first about cytokines and how they are targeted for treating patients with IBD. Of course, we already heard about ustekinumab in one of the previous studies, so I'm not going to spend a long time talking about it. However, we have seen how the results of UNIFY was very impressive with positive results indicating that ustekinumab is effective as a treatment option for Crohn's disease for both induction and maintenance. As you can see here, patients who received induction followed by 90 milligrams every 8 weeks as maintenance after almost a year of treatment did fairly well, with about 53% in clinical remission, which is quite remarkable if we look at previous failure rates in patients who were treated with other options. Now, of course, this was also mentioned that all of the reports so far seem to point that Stelara or ustekinumab is a fairly safe drug with a very low risk of adverse events or even neoplastic complications. Of course, subsequently, the drug was examined in ulcerative colitis, as we've heard, and the drug in the study UNIFY, and the study met the study outcome statistical significance as the primary endpoint clinical remission, in addition to multiple secondary endpoints, were all positive in the induction and maintenance periods. This had led to the development of other drugs in the same platform of medications. As you can see here, the IL-23 inhibitor risankizumab was also studied and proven to be effective for Crohn's disease. It's now also in the late stages of development and approval, and will probably be available as a treatment option for Crohn's disease, in addition to many more medications in this category. We also heard about vedolizumab by Prof. Izzat, which is a drug that was developed in the mid 2000 with multiple studies in phase 2 and 3, and eventually in phase 4. The drug is a gut selective agent that works on inhibiting leukocyte trafficking into the gut, i.e. Eliminating inflammation locally rather than systematically. Here is the a cartoon that shows how the drug works by inhibiting the interaction between alpha-4 beta-7 and MAdCAM, which Professor Azad has also highlighted in the previous lecture. We're all familiar with the study Gemini, or the Gemini series of studies. We know Gemini 1, where patients were studied who received vedolizumab versus placebo, and all of the primary and secondary outcomes and investigational outcomes were positive for this drug in both patients who were anti-TNF naive or anti-TNF exposed. Of course, the drug went on to be examined in Crohn's disease and later on in multiple other indications such as fistulizing Crohn's disease as well. Systematic reviews and meta-analysis have all summarized and pooled data from all of these studies supporting that the drug is fairly effective with a very significant chance of success for induction and maintenance. And again, this is a drug that is proven to be very safe with virtually no or minimal risk of side effects, the commonest being nasopharyngitis that sometimes occurs with patients. But other side effects such as infection and neoplastic complications are highly unlikely. Of course, other gut selective agents have been developed that work in different mechanisms through inhibiting other interactions on the leukocyte gut vascular barrier, and some of them have already in the late phases of development, as you can see here with anti-MAdCAM from the Trundot phase 2 study for ulcerative colitis, which again is in the late stage of development and is proven to be effective. Etrolizumab, which is a Rumab beta-7, is a drug in the late stage of development, which has been studied for moderate to severely active Crohn's disease. In this study, as you can see, patients were examined who received placebo or etrolizumab 100 milligrams sub-Q versus 300 milligrams sub-Q. And you can see how the population who received the 100 milligram dose did fairly well, both the anti-TNF naive and anti-TNF exposed populations. So we have a pipeline full of medications, biologics specifically, that are being developed and are in late stage of development. Some of them with an imminent date of launch, all working on the leukocyte trafficking mechanism of action. This is the galaxy, the CD galaxy, where we can see different drugs being developed, whether they are cytokines or leukocyte traffic inhibitors, or even other mechanisms of action. Again, for ulcerative colitis, we have several drugs in late stage of development. So we can see that the future is hopefully bright. We expect that over the 5 years, many of these drugs are going to be released, and hopefully we'll have many options to choose from when we are about to start a patient on treatment. And hopefully with time, we can find a drug that significantly interferes with the disease course and prevents complications with time. So in summary, the take-home points: there is a large unmet need for effective medical therapies for IBD. Anti-TNF agents are effective therapies for IBD and can be used at different time points. Gut-selective drugs that are associated with a lower risk of systemic infections are becoming more attractive options. Other forms of biologics such as SLAMIs and anti-cytokines are new biologics that can be used to treat IBD. Thank you very much for your attention, and I'm happy to answer any questions.